panel event today is going to be bringing us all together to discuss the growing field of non-animal methods or non-animal models. And we have a lot of really great information to go through today. So I'll just go ahead and get started. We'll be exploring non-animal methods or non-animal models, NAMs. We'll be going over the key NAM categories, the regulatory process, case studies with Lorna and Tara here, challenges, and the road ahead. We'll be moving through these slides with some discussion prompts for panelists, and then we will move into the more the open Q&A at the end. Everyone feel free to use the Q&A portion to communicate and ask any questions that you may have as we go through the presentation. Today we have Dr. Lorna Ewart, who is the Chief Scientific Officer at Emulate, which is a biotechnology company pioneering work on organ chips. And you will learn more about that today. Dr. Tara Barton-Maclaren, who is a Senior Research Manager in the Prioritization and Emerging Science Division at Health Canada, the Government of Canada, and Troy Seidle, who spent 20 years with Humane World for Animals as Global Vice President for Research and Toxicology. And now Troy is the President of the Animal Alliance of Canada Fund. And Troy is also a member of our Animal Alliance Advisory Group on Humane Science. We are here today to discuss non-animal methods in research, teaching, and testing. There will be some examples. So if anyone has any questions or if you see any of the categories come up and you're unclear, we're here to talk about that today and demystify NAMs of non-animal methods and explore what is possible without using animals in science. Animal testing has been the default for the past century, and now there is growing scientific and regulatory momentum towards alternatives. And so we want to provide more information about that, and that's why we're here today. So what are NAMs? There are quite a few terms. You may have heard the term new approach methodologies. You may have heard non-animal methods as we're referring to it today. And we will go through that with you. The definition, as we see it on the Health Canada website, is that NAMs are broadly defined as any technology, methodology, approach, or combination that can be used to replace, reduce, or refine animal toxicity testing and allow for more rapid or effective prioritization for the assessment of chemicals. It is an umbrella term. It covers a wide and evolving range of tools, and advances in biology, computing, and data science have made non-animal methods increasingly viable, and we'll go through that today. The limitations of animal models has been and is widely discussed. Regulatory bodies themselves are acknowledging these limitations. And so I'd like to jump in and start the discussion. And this question is for all panelists. But what failures or problems have you seen firsthand with animal models? And anyone can take that question and we'll pass it along.
Sure, I'll jump in. Looking back over the decades, you see some animal tests that have been repeated multiple, sometimes dozens of times, same chemical, same procedure, different results. So whether we look at lethal dose tests from the 1920s, rabbit skin and eye irritation tests from the 1940s, these are really old procedures, but they've never been validated for their human relevance or their repeatability. So even if you're trying to compare a rabbit to rabbit from one lab to another, you get a tremendous amount of noise there. And that's not even trying to then make the leap to you or I, what's going to happen to us with our unique physiology. So we just see this again and again with animal tests when you have the luxury of the time and you see these repetitions happen. So that's just one of many examples, but I'd love to hear from other panelists.
Sure, I can add to that. Thanks, Troy. And I think from the context that I'm coming from, one of the limitations is also a practical limitation, and that being one of general data coverage. And I think that really touches to the limitation that you're seeing on the screen here in terms of the high cost, long timelines, resource intensity also to generate this information. So for the chemicals that we're interested in, those are chemicals that are already on the market in Canada. That means that many of these chemicals remain insufficiently characterized. And so this practical limitation also turns into a scientific limitation in that it creates some uncertainty and the potential for blind spots in some of the assessments that we're doing. So thinking about some of the limitations, I think we could turn it really to the opportunities where the non-animal methods or the NAMs can help us to improve understanding. And I think Roy touched on one being that human-specific biology or better understanding human relevance in terms of variability, susceptibility, mechanistic understanding of toxicity. But I think it's also quite well recognized that there are some toxicities that are not well covered or not well characterized in animals. And a couple that we're thinking about are, metabolic disease and cardiotoxicity, for example. Um, I have one example that I just wanted to highlight here of some work that we'd been doing collaboratively under the Accelerating the Pace of Chemical Risk Assessment. And this is a paper that was published in August last year, where we really tried to better understand this question on limitations and took a look at both quantitative and qualitative concordance between non-clinical, so the animal toxicity data, and clinical human data, as well as in vitro data from ToxCast database, and again comparing that to the human. And I think that that deep dive, really provided us with some additional insight where we found when we were looking at the animal studies, they really only show a moderate quantitative agreement in terms of protective power. Looking at the animal against the human, we found that in those data sets that we looked at, they didn't really perform all that well at predicting what toxic effects we would find in humans. But interestingly, by comparison, the in vitro studies were comparable in a quantitative way and offered that better understanding of mechanistic relevance. So I think, even looking at those comparable data sets, there's some really important take-home messages that, when we were looking at the in vitro, the non-animal data, there was similar quantitative performance in terms of that dose at which we can anticipate effects or below which we can anticipate effects not to happen, but different strengths. And so I think this is one example of how we're working to better understand the limitations and or the opportunities of where we can introduce non-animal methods into our thinking and into our work.
Yeah. Maybe I'll just finish up and build on what Tara and Troy have already said here. My background, of course, is pharmaceuticals, so I'm going to come at it slightly differently. And first of all, talk about the toxicology aspect and then think about efficacy. So, just really bringing home that concept of human relevance. There's a really famous example regarding the consequences of misinterpreting or incorrectly judging risk assessment based on animal data. And it comes from a study showing that an antiviral drug known as thiouridine actually went through its typical rodent study as would normally be seen to progress first time in human, and no flag was raised. By the time it did make its way into the human population, it actually resulted in the deaths of 5 clinical trial participants. So that, is clearly shocking, and it's absolutely what, one would try to avoid during the drug development process, and in making that transition from the preclinical side into the clinical side. And hence there is a great deal of conservatism, and rightly so. But subsequent work is showing that actually what was happening was at in the liver, we have what's known as drug transporters, and there were species differences in these drug transporters, such that in the human, the drug was accumulated within inside the cells of the liver, the hepatocyte, whereas it was not in the rat. And it was that accumulation that actually ended up leading to the tragedy that I've described. So I think that's a very real example where we know in terms of toxicology that the lack of human relevance can actually be problematic. Um, if we think about efficacy, I've spent a chunk of my career also on the efficacy side, and particularly thinking about oncology. And again, quite often what's used there is a xenograft model where the patient tumor is actually situated on the back of the animal, and they're simply measuring the size of that tumor. And really, what you can do over time in most mouse studies, these tumors disappear entirely. And so, hallelujah moments. If any of our mice ever were to get cancer, we could probably cure them. And I use the word cure effect, properly in this context. Yet many, many, many of those drugs that transition into humans, of course, we can't use the cure word as readily as we would want to in the human situation. And again, there's a really nice study published looking at insulin growth factors, and trying to target those as targets for oncology, and across 16 different molecules targeting that family. They've shown that preclinically cancer disappeared. In the clinic, it had no clinical benefit. So again, just highlighting two published examples in two different arenas, and why, as a drug developer, it's very interesting to me to consider not just the limitations of the animal models, but also think about the human relevance of some of the new technologies.
That was great. Thank you all for your thoughtful replies. And the term human relevance is one that we use often and we've had conversations about, and it's a very important key term that I'd like everyone watching this to keep in mind throughout the presentation as well, human relevance. So moving ahead, the the scientific landscape and the key NAM categories. So, Troy, I was hoping that you could walk us through the categories that we have listed here to get us started.
Sure, happy to. We're going to keep this pretty high level, but when we think about the toolbox of NAMs, and especially when we hear the word new approach methods, I think an important realization is that a lot of what is available today isn't new. I was at the table at the OECD negotiating for the uptake of human cell-based three-dimensional skin and eye tissues as global guidelines back in the year 2000. So 26 years ago, we achieved global acceptance of these. They've been available years before that and going through validation studies. So some of this stuff goes back that far. We have some genetic toxicity tests using salmonella bacteria that goes back to the 1980s and before. So a lot of these methods have been around with us for quite a long time. There is a high level of comfort with them, which is the good news. I think some of the more challenging news is that whether these tests are still fully replacing the use of animals consistently across sectors, across countries, that's a work in progress. But, really solid good news, starting at the top with the in vitro methods, particularly our ability to shift from using rabbit tests for the skin and eye types of effects, skin allergy tests, to the use of these particularly human-derived cell-based methods. In silico is computer-based. So we have models that are built and trained using legacy toxicity information. And these are, really giving us the power to do instantaneous predictions. And they can be quite accurate depending on the quality of data that's used to program them. So you have these structure-activity relationship models, which are looking at the molecular structure of a compound and correlating that with its biological activity. And these are, again, plentiful within the toolbox of methods that are available. Typically, what we're seeing, though, is not a one-to-one replacement with many of these. We're using these within these integrated strategies where you may have two or more methods that are required to really fully recapitulate what's happening within a more complex type of biology. The microphysiological systems is Lorna's wheelhouse, so I won't say too much, but more to come when we hear about organoids, organ on chip, or even these integrated body on chip models that are linking up more of the organs, organ systems. Omic approaches is zooming right down to the molecular level, looking at what happens at the level of the genes, the proteins, the metabolites when they're exposed to different chemicals. And then adverse outcome pathways it's a way of picturing the complexity of human, other animal biology from the molecular level to the cell, tissue, organ system, the full organism. And rather than looking at it in a very reductionist, compartmentalized approach, AOPs give you the full landscape, zoom out approach to be able to then understand if you see an effect in the molecular level, does that actually cascade into something that's harmful, or does the body adapt? So AOPs really help to take the glut of information that can be available from some of these molecular tools and contextualize them within a more relevant whole body type of picture.
Okay, fantastic. And I was going to ask, we are going to go into this, but I'm curious how mature, and this is for all panelists, how mature are any or combination, any one or combination of the categories listed here, how mature are they for regulatory submission today?
Yeah, I don't mind jumping in here and just again citing something from the literature. Again, bias here towards drug development. It was published at the end of last year by two of the representatives in CEDAR, which is the Centre for Drug Evaluation Research in the FDA. And they looked at IND submissions. So those are investigational new drug applications, which is where a sponsor is asking a regulator to transition from the preclinical space into the clinical space and presenting a case for that to happen. So having made a risk assessment. So using those documents, this group, they evaluated all of those INDs that they had accumulated over a 15-year period and actually looked for submissions that had in vitro, in silico, in chemical, non-human, in vivo. That's where their sponsors are using a phylogenetically lower animal, something like a Drosophila, for example, would fall into that category. And then just the other was just the catch-all for everything else. And what they found was that actually 93% of these submissions contained some of these approaches, and they were able to carve that into sort of two main buckets. So this speaks a little bit, I think, to the maturity. So 49% of those were actually in silico methods. So again, my bias and background a lot is in liver. So there's been a number of in silico tools around that are highly characterized now that allow a sponsor to understand potential metabolite generation by the liver, and metabolites can become hepatotoxic molecules. So although the parent drug itself is not hepatotoxic, the liver can make it such due to its role. It is there to metabolize substances that it doesn't recognize to help eliminate them from the body. So there was 49% of applications that did include an in silico method, highly skewed to understanding metabolite identification. And then 44% of them actually did also include in vitro models, and that those were predominantly in the stem cell area and more simple in vitro methods, 2D cultures, so perhaps more akin to the top bullet there. Now, they weren't seeing 3D models and microphysiological systems. I think that reflects the fact that technology really was discovered around, in 2010, 2012, with many companies starting to commercialize around 2018 to 2020. So I should imagine if one were to look at submissions, maybe again in 5 years' time, I think we might see a different picture. So all of that's to say, I think it's a maturing field for sure. And we will get into talking about that later. I'm almost certain in this presentation here, but hopefully those are useful data points to start this discussion off.
Yes, that's fantastic. And I should mention, for the audience today and for anyone watching the recording, the resources or any references to studies, we will make those links available so that everyone has a chance to look at that in more detail as well, if you're wondering throughout the presentation. Thank you, Troy and Lorna.
Great.
So,
Oh,
oh,
I
sorry.
was going to say, did you want me to weigh in? I was just going to kind of echo
Yeah.
really kind of underscore the message that, despite the fact we've called them new approach methodologies, Troy said conceptually they're not new. And, even within the Canada's Chemicals Management Plan, a number of the NAMs that have been mentioned here have been essential for us delivering on our mandate since initial inception of the CMP in 2006. And so, in particular, the in silico methods have contributed to prioritization, categorization efforts throughout the various phases of the CMP, including read across is one perhaps that wasn't mentioned, but can be kind of integrated into this computational in silico approaches. And I just wanted to highlight two other areas. The defined approach for skin sensitization is something that was recently published as the first defined approach to become an OECD test guideline a couple of years ago, combining in silico predictions and chemico and in vitro data. And we are seeing increasing submissions of that type of information in our new substances program. And in the pesticides context, a more mature, I guess, area is the acceptance of the in vitro data for estimating dermal absorption. And this replaced the triple pack. So now accepting the in vitro data only. And I really think that in terms of maturity, for regulatory submission, we do have to think about the differences between the many legislations. And sometimes maturity or acceptance really is a function of validation or establishing that scientific confidence. So that is something that we're constantly thinking about as the methods continue to progress. We're introducing increasingly complex NAMs or non-animal methods. And I think, Lorna's example is really nice when we're thinking about, the progression in terms of the introduction up to the microphysiological systems.
If I can jump in and build on Tara's point, I think there are a couple of other concepts at this stage that are worth introducing, which one of them is the idea of a waiver. So you have the information
Mhm.
requirements, and sometimes the answer is we don't need a NAM. Sometimes the answer is we don't need the study, full stop.
That's
So
right.
we can waive conditionally, or in some cases, we're looking back at tests, for example, in the biologicals and vaccines area, you have one called the abnormal toxicity test, which is looking at just abnormal effects, fishing expedition for human outcomes. And you have a target animal batch safety test for veterinary vaccines. And broadly speaking, those are viewed by the pharma industry and by relevant regulators as unnecessary. So those tests are increasingly just being dropped and being removed. So we have some of those. There's an example
Mhm.
in the pesticide world, a one-year
Yes.
dog test, which across the globe, it took nearly 15 years, but we were able to eliminate that worldwide. So you have waivers and deletions of tests that you learn by experience we simply don't need. There's also kind of where is this going? The concept of next generation risk assessment, which is similar to the way we do things already today, but taking inputs like exposure assessment, are we actually going to be exposed? If so, under what conditions? What information do we need really to make a safety decision in the real world, rather than here's a list of animal test requirements, or here's a list of safety questions, here's a list of animal tests, call the testing lab and do the tests. And that's the traditional approach, but really being able to reorient to ask more thoughtful, more strategic questions is really the direction of travel. That's the opportunity for the future. We are not there yet, but again, we do see some regulatory authorities, including Health Canada, who are really at the cutting edge of that. And how can we start to implement that in practice?
Fantastic. Thank you all. As this is the perfect time to transition, Troy, the regulatory process and acceptance. So I'll have you take us through a general overview, and then we'll move into the Canadian context as well. But Troy, do you mind explaining kind of where we're at across the board with the regulatory process?
Sure. So bad news first. Um, Look around the room you're in. Look at the computer you're watching this on. Virtually everything we come into contact with in our daily lives has been animal tested at some point in the past. That can be the plastic ingredients in the computer, the adhesives, the fabric dyes, look in your medicine cabinet, you name it. And that is what I think of as the wrong thing for the right reason. We legitimately expect safety. We expect that someone is watching our backs. We don't typically think much more about what that process looks like. We don't look under the hood, most people. But the truth is, regulations exist for drugs, for vaccines, for pesticides, industrial chemicals, food additives. And, again, this exists for a good reason, for human health and environmental protection. And these are government regulators doing their job, looking at health and environmental effects before substances are released to consumers or to the environment. The way that the system normally works is the questions, is this safe? Is this effective? Are laid down in regulation. The way the questions can be answered is laid out in testing guidelines. So if we're looking at testing guidelines for cosmetics, chemicals, pesticides, those are typically under the OECD, the Organization for Economic Cooperation and Development. For pharmaceuticals, the human is ICH, veterinary, BICH, and you have pharmacopoeias for the more biological kinds of products. The good news, it's again a cost benefit with all of these. The idea of having internationally harmonized guidelines was intended to avoid redundancy, where different jurisdictions have different requirements that have to be repeated. So the idea of mutual acceptance of data has reduced duplicative animal testing in some cases. The flip side, however, is that these methods can become frozen in time, and having 35, 38 OECD countries agree on a method is hard enough. To get it on the books, getting it off the books is nearly impossible. I've seen it happen 2 or 3 times in my career. So that's the really double-edged sword of this. We have been successful in having around, I think it's over 40 non-animal methods taken up in OECD test guidelines over the decades. So it is good news that the NAMs are taking their place alongside the animal tests, but it's still, a question of how do we transition from an obsolete method, or when do we acknowledge that a method is obsolete and needs to be moved away from? So some milestones that are worth acknowledging. Over the past decade or more, we've seen a tremendous worldwide shift in the cosmetic space, really a cruelty-free makeover, you could call it, where now 45 countries and counting have outlawed the practice of animal testing for cosmetics. And that's really changed the narrative from can we move away to how quickly? And that's really helped to frame some of the conversations in other sectors. And it's not about banning necessarily, but it's proof it's proof of concept that we can move away from the old. We can still assure human safety. It's not about trading animal safety for human. And I think that's been a really productive realization for both companies and regulators that it's not a trade-off. It's not your child or your dog. We're also seeing increasingly legislation, the policy language coming from politicians that is specifying concepts like animal testing as a last resort. Whether that is applied effectively in practice is another question, but it starts with the words on the page. And we are seeing now a trickle-down effect where at the level of the testing guidelines, you're seeing increasingly there's an uptake of NAMs and there's a waiving or deletion of the obsolete animal tests. And then the last and most recent piece within this development, we're doing a really quick flyby, is the development of transition roadmaps, where at the national or agency levels, we're seeing really intentional, thoughtful plans coming together to speed the uptake of NAMs and, as much as possible, have plans, some strategies for eliminating the corresponding animal tests.
Now, moving ahead into the Canadian context, Tara, I'd like to invite you to explain a little bit more about this to us.
Okay, great. Thank you. So it certainly has been a busy and exciting time in the context of Canada's regulatory landscape over the last couple of years, across different areas, across different legislations. So in 2023, cosmetic animal testing was banned with support from Health Canada. And this marked maybe one of our first more significant policy shifts. So in this case, the amendments to the Food and Drugs Act, or FDA in Canada, included prohibition of testing of cosmetics on animals in Canada. Also, the selling of cosmetics that rely on animal testing data to establish the product's safety, and included prohibiting false or misleading labeling pertaining to the testing of cosmetics in Canada, or on animals, sorry, in Canada. Now, under this, there are a number of alternative approaches that can be used. I won't go through them. If we kind of get to that in conversation, we can. But this really set the stage for the additional actions then that have been taken. So the area of chemicals regulation is the area that I work in day to day. And so we also had a modernization of the Canadian Environmental Protection Act, or CEPA. This is the act under which the Chemicals Management Plan is for the regulation, the assessment and regulation of industrial chemicals. And this modernization also in 2023 included the addition of animal protective language. In response to the new requirements that were added under CEPA, Canada developed and published a strategy in 2025, which will help guide and promote the use of the scientifically justified alternative approaches to replace, reduce, or refine the use of vertebrate animals in toxicity testing whenever possible, particularly under CEPA. So this includes activities for both our new and existing substances programs in Canada. And I've outlined in this slide on the right-hand side the 5 key elements that the strategy outlines. And so kind of looking from the top, and we intended these to be really presented this way to reflect that the goal is for each of these activities to be ongoing in an iterative and progressive way. First, really looking to identify and prioritize NAMs that specifically are able to address our regulatory needs. So thinking about what are our requirements within our different acts and legislations. And then working to advance the NAMS research and the data generation, being able to, look back and address some of these regulatory needs and requirements. Critical to the success, in the past and in the future is being able to promote and establish harmonization. So we want to be aligned internationally with the advancements and the use of the non-animal methodologies, and do this through ongoing partnerships and collaboration. One of the cornerstones to our program in Canada is also the engagement that we have with our partners and stakeholders. And so certainly this will continue to be a very important element of the strategy moving forward, with the ultimate goal to be able to implement in CEPA regulatory programs these non-animal methods. And although this strategy, as currently published, is being developed under CEPA. The aim is that it could be applicable where relevant and possible to other Government of Canada regulatory programs. And so, also as noted on the slide, there are many groups within and beyond the health portfolio that are engaged in activities related to NAM's development and use. So in particular, some of the waiver activities were noted. This is quite specific to our Pesticides Regulatory Directorate, formerly Pesticides Management Regulatory Agency. We have activities ongoing with the Health Products and Food Branch. And of course, especially in the context of our Chemicals Management Plan, this is done in collaboration with Environment and Climate Change Canada. So some activities ongoing there in the context of environmental and ecological health. Yeah, so I think that kind of sums it up just in terms of those core activities that have happened and are really guiding our direction moving forward in our regulatory work.
That's great. Thank you. And we'll go through some case studies as well. So Lorna, I'll turn it over to you to walk us through the next two slides, and then Tara will turn it over to you.
Yeah. Thank you, Bianca. So what we're looking at here is work that we've been doing at Emulate over the course of a number of years, actually. And maybe I'll just start on the sort of top right-hand corner, the cartoon that's there. So for those who are not familiar with Emulate and what we're building, we're building what's called liver chips. So we're in the flavor of microphysiological systems, organs on chips. And our chips essentially are about the size of a computer memory stick, so about this kind of size. And they're designed really with interfacing engineering and biology. So what we mean by that is the design of those chips contains two parallel microfluidic channels. And that's really where the engineering comes in. We have what's known as a pressure-driven flow, and this allows us to start to expose cells within these chips to what would be like almost like a circulation if we were thinking about the body. So all of our organs are continually perfused. So at Emulate, one of our guiding principles is that by the constant perfusion of cells in cell culture, as well as additional mechanical forces. So for example, in the intestine, there's a motion called peristalsis, which is what moves the matter through the intestine and ultimately to be removed from the body. The cells are experiencing lots of different forces, and we can recreate those forces in our chips as well. Now, the liver, it's a solid flat organ. It doesn't really have any, fixed mechanical forces in the same way as an intestine would. So we don't use the stretch function when we're thinking about the liver. So what we're really thinking about is the simplest functional unit of the liver, and that's known as the liver sinusoid. And to recreate that, then we take the most abundant cell in the liver, which is called the hepatocyte, and we place it on the top channel of our two parallel microfluidic channels. And then we use there's a membrane that separates these two channels. So we're using the membrane to recreate another part of the anatomy known as the space of Dease. And then in the channel below, what we're doing is we're seeding the Kupffer cells, which is an immune cell, the stellate cell, which comes into play when we're thinking about fibrotic responses, for instance. And then we have the liver sinusoidal endothelial cell, which are those big kind of disc-shaped cells that you can see in the cartoon as well. So this is 4 cells. They're all human cells. They're all human primary cells. And at the moment, all of our cells usually come from different donors. And what we wanted to do, so that took us many years to build, but what we wanted to do is we wanted to perform what at the time was the largest study that had actually been performed in these technologies. So we wanted to look at how well this model performed in its ability to predict drug-induced liver injury. I alluded to it earlier, but for those who are not in the pharmaceutical space, drug-induced liver injury, or DILI for short, is actually one of the leading causes of drug withdrawals or labels, black box warnings, restrictions on how medications can be prescribed, etc. So it is a problem, and we wanted to see how well our model would work in terms of predicting it. Now, as we were actually thinking through our experiments, again, a lot of things come through serendipity, and there was some industry guidance produced by a consortia of pharmaceutical companies known as the IQ MPS affiliate. And they publish some guidelines that are helpful for organizations like Emulate who are building these models. And they said if you're going to build a liver to look at drug-induced liver injury, it needs to look like the following. So there was a set of criteria, and what we essentially did was we walked our liver chip through those criteria. And this culminated in testing a set of molecules. We tested 27 small molecules, 22 of which were known to cause drug-induced liver injury in the clinic. So all 27 had been in the clinic, but 22 of them were positive for drug-induced liver injury, or DILI. And what we found, if you look at the bottom there, the big 87%, we found that our chips across 3 donors, and with the team being blinded to the identity of these molecules, the liver chip was able to correctly identify 7 out of every 8 hepatotoxic molecules. And that gives that model an 87% sensitivity. And as I mentioned earlier, there were 5 molecules that were clean, truly clean in the clinic. And the liver chip saw all 5 of those as being clean. So it gets 100% in terms of its specificity. Now, within that set, we were also able to compare against literature data from a different type of in vitro model, hepatic spheroids. And we found that against the similar compound set, hepatic spheroids really were no better than chance in terms of predicting whether a molecule would be toxic when it got into the clinic. And we actually then also thought about the animal studies. Now, we didn't undertake any animal studies, but what we know, and my background for 12 years, I was a project toxicologist in industry. What we know is that to go into humans, animal testing is still a requirement. And we know for small molecules that they're tested against usually a rat and usually also a dog. So knowing that these drugs have been in the clinic, what we said was, well, animals have a 0% sensitivity. Now, what we're not saying that they didn't see the toxicology, but what we are saying is that if they had seen the toxicology, there may have been a different decision by the sponsor and or the regulator to progress that into the clinic. And of the molecules that we tested, many of them were associated with patient fatalities as well. So we would attest that actually the assumption of whether the animals saw it or not, the risk assessment was still inadequate. Now, within this data set, we were able to look at what's known as structural analogues. And now, again, if you're in the pharma industry, what you're trying to do is progress chemical series through. These chemicals all look quite similar to each other, but sometimes it's just the addition of, let's say, a chlorine atom, for example, into the molecule that might lead to a toxicity. And the match pair may not have that chlorine, and therefore it may be clean. So there were 7 matched pairs within our data set that we tested. And again, our liver chip was able to spot all of them. So it was able to spot the more toxic and the lesser toxic of those analogues. So actually, this data, we tested across 870 of these liver chips. It was quite an undertaking by my team. It took 16 scientists, 16 weeks to generate the data. And that actually allowed us to progress into the FDA qualification program known as iStand. And maybe, Bianca, if we move to the next slide, then we can talk a little bit about that. So there's been quite a lot discussed now by the US FDA, including actually some guidance documents on validation that have just been released as well. And so it's important. I think we did have it on a prior slide, but there's the definition of validation is different to qualification. And I'll go with the US description because it's the one that I'm most familiar with. But validation really talks about the reliability and the reproducibility of a model. And that's really important if someone's going to use that model to submit data into this document that we talked about earlier to allow a regulator to make a risk assessment. By going through an additional step of qualification then is looking at actual performance of that molecule. It's going into the nitty-gritty, in my case, thinking about flow rates, how have I qualified my cells, how have I assumed or shown that the endpoints that I'm measuring are actually clinically appropriate and themselves have some validity as well. When a tool is qualified, then it can be used by any sponsor within a particular context of use. So there's a subtle difference between validation and qualification. We want to be regulatory qualified. So we submitted the data I just described in the prior slide to the FDA, and we were the first organ on a chip company to be accepted into the program. There are about 22 letters of intent that have now been accepted. It's a 3-stage process, as you can see in the bottom there. And many of those actually are organ on a chips that are now in the program, which is fantastic to see. It will also, of course, take in silico tools as well into this particular qualification program. The second step then is just to say how you're going to complete your qualification process. That's known as a qualification plan. We're the only company in the organ chip field to have had that now accepted. And right now, my team are just completing that qualification exercise. And we want to submit our full qualification package this year, which after about a 10-month review, which is where the FDA are publicly committing to right now, we should be qualified. So fingers crossed, if anyone invites me back this time next year, I'd love to be able to tell you that we've got our tool qualified. So hopefully that gives a little bit of a flavour about the process and some of the data that goes into supporting these types of qualification exercises.
Thank you so much. I'm going to save my own questions until the Q&A section so that Tara can go ahead. I'm sure I'll have many more, but thank you for that. So Tara, I'll just turn it over to you and feel free to let me know when to toggle and I will move over to your second slide.
Okay, that's great. So intentionally, I wanted to switch gears a little bit from the lovely work that Lorna showed and the in-depth amount of research that goes into the qualification validation of each particular method or assay, if you will, and kind of bring it into a higher level view of then how do we think about actually using the data that comes from these methodologies. And so as a data user, one of our primary goals is to continue to support the interpretation and integration of information in a very transparent and consistent way. This is the way that we typically handled the traditional studies and the way that we want to continue to be able to integrate the NAM type data. And so to support our prioritization process, we developed an automated workflow, which helps us be more efficient and very consistent and transparent in the way we, collect data, interpret data, and prioritize the chemicals that we want to look at further. And so using an automated approach, we were able to collect millions of domestic and international records. We have the ability in a more, I guess, a rudimentary way, but be able to identify where the data gaps are, be able to implement novel consensus modeling approaches. So this is where we're integrating a lot of the in silico computational models, be able to apply structure-based automated read across, again, looking to see where evidence already exists to fill gaps that we have. We're able to, in an automated way, reconcile differences that may be observed across data types. So we want to be able to look at all types of information and understand how they complement or how they're different and be able to do deeper dives to understand those differences. And then at the end of the workflow, we have a hazard and exposure based prioritization scoring. So within this workflow, consistent scoring criteria are applied to rank the substances based on exposure and hazard indicators. And this generates a nice report so that our evaluators can then put an expert eye to the information that we've accumulated and evaluated. The next slide, please, goes into a little bit more of the details and maybe the complexities of exactly what this tool is. So the tool is developed in KNIME, and this analytics platform is really useful because it allows us to maintain this modular design, which inherently has flexibility to continue to adapt to evolving science, emerging data sets. It allows us to integrate various IT platforms and coding languages. So as new information becomes available, depending on where that publicly available data set may live, we're able to integrate it into, kind of the dynamics of this workflow. It is intentionally designed to be able to collect and interpret new approach methodologies alongside with the existing traditional data streams. And in our context, I just really want to highlight that our risk assessments are based on publicly available information. In the existing substances program, we don't have specific data requirements, so we do not require any type of testing. We're using the testing or the data that is available internationally. The way we've designed this workflow is that it can combine various biological levels into a decision framework. So this touches back really on what Troy was introducing in terms of the different types of NAMs, where we're really thinking about each of these hazard-specific nodes as an IATA, an integrated approach to testing and assessment. We're thinking about how can the AOP framework inform how we organize and interpret the types of information that are available to us. So as you can see, kind of along in the blue box, what we're calling our hazard-specific modules, we have the opportunity to integrate all available information to address the different types of toxicities that are critical to our risk assessments. We are also looking at how we can improve kind of our efficiency and reproducibility when we're looking at the emerging science. And this really is falling into right now we're calling it more the hazard or the agnostic or protective not modes, where we're looking at high content, high throughput type in vitro data. And so one example is ToxCast bioactivity. We have a workflow here for the interpretation of that type of information. We've published what we're calling a science approach document. And so what I'm showing you here is also published as a science approach document. These documents are basically the objective of these documents is to be able to describe how we are using two new types of information, their interpretation, and their proposed context of use. The reason we publish these new approaches within these types of SIADs, as we call them, is it gives us an opportunity to engage with our stakeholders. Each of these documents are published for a 60-day public comment period, and it allows us feedback on, whether it be the data, the interpretation, the proposed context of use, etc., which helps us to evolve these also in an iterative way. The other area that we're actively working on is the area of transcriptomics. We are currently working on enhancing this mode, and what we want to do with this is to develop a stepwise workflow that allows us to capture the processing and handling of the high throughput transcriptomics data in order to derive points of departure from available in vitro data sets. Through this process, our goal is to be able to demonstrate the automated capacity of the workflow to scope and evaluate chemicals for use by the risk assessment community, and also necessarily be able to provide a consensus on what we propose to be the most practical approaches to be able to derive these in vitro points of departure using this type of in vitro data. So these are examples of how we are setting the stage to be able to incorporate the emerging types of data set in our day-to-day work. The other area, if we go back up to the hazard specific, so, kind of that larger workflow in deriving that bioactivity point of departure is a more generic way of looking at the information, but we're also exploring ways in which we can use the in vitro data to more specifically predict or address toxicity endpoints of interest. And a couple of areas that we've included this type of information is in the endocrine screen, so the EDC screen on the far in the yellow box here. We've been able to incorporate the ER and AR models. We've been able to incorporate the ER alpha biomarker, which is from the transcriptomic data. So all examples of how we're able to use this information also in a predictive way to inform some of our assessment activities. A little bit similar to the example that Lorna showed us, there's also a biomarker that we can incorporate now into the mutagenicity screen. And this is a biomarker that was led by Canada. It's just completed a multi-ring trial, also funded through FDA Drug Development Tool Program in partnership with HESI Global. And this is a toxicogenomics biomarker for a DNA damage inducing agents. And so there's another example of when that information becomes available, we're prepared to be able to integrate this into kind of these integrated approaches, and really be able to deliver decisions through this evidence-based integrated knowledge.
Fantastic. Thank you so much. We are still doing well for time. We still have our time for questions, but I will move us into challenges and the road ahead because I did have a question and a follow-up question here for all panelists. And I think this will transition us really well to our Q&A portion. I know there are some questions coming in now. When thinking of the challenges and the road ahead, which challenge, and it doesn't necessarily need to be listed here, which challenge is the most urgent to solve in your opinions? And what could the audience do or someone watching this recording do to help support the transition? If you could answer that, if possible.
I'll jump in. It's hard to prioritize one, but I would say, if we start with inertia, it's really difficult once somebody feels that a method it's available, it's familiar, to replace it or waive it with something that is new or unknown. That is, human nature does not like that. None of us do. And that's tricky to have that conversation one time with a group of regulators and companies in one country, but realizing that this is not a country by country approach. This is not a why doesn't Canada ... It's global. This is a global trade situation. This is global science policy. And we have bookends. There will be an early adopter, someone who goes first, and there'll be somebody who goes last. And as long as the bookends are far apart, we really need to be able to pull them closer together and have this conversation as a global team to problem solve and say, again, just ground ourselves in what are we trying to predict? Human, rat, fish? Um, let's have the scientific conversation. What does the evidence support? And then it's all the things you might not expect. It's the yeah buts, the uncertainties, the confidence, the fear of making a mistake, and the different legal consequences in certain countries, which can be down to an individual regulator going to jail. I mean, it can be quite severe in some parts of the world. And if I'm a company and I'm trying to sell whatever it is across the globe, the lowest common denominator is such a powerful thing because it's the path of least resistance. And that really, from the corporate, from the regulatory, legal points of view, really favors the status quo. And it doesn't favor the bold, it doesn't favor innovation whatsoever. So really being able to crack that inertia, and that is tied to the risk aversion piece. What is the evidence base? But yeah, I think the biggest barrier these days is less scientific. It's more, it's human. It's human beings being human beings. And although that is a challenge, I think that's also the solution. Being able to communicate with each other, being able to find that common ground, build the trust, build the confidence is the only way we're going to get out of this.
Yeah, and,
Yeah,
and maybe just, oh, go on, Tara.
oh, are you sure? Sorry. And I was really going to jump on that and say there one of the core challenges, kind of building on what Troy has introduced, is really what we might call a decision gap, where regulators need the NAMs to support defensible quantitative risk assessment decisions. And I think there is still a lack of that standardized translation of widely accepted NAMs that can be converted into outputs that address the regulatory metrics, whether it be the equivalent to a NOAL or a LOAL, an adverse outcome. And regulators are really still grappling with how NAMs fit into frameworks like the adverse outcome pathway, for example, where earlier events or indicators are reflective of an adversity that it was likely to happen at a given dose. I think there is also an integration challenge. Individual NAMs are very strong, but we're looking at different types of NAMs as various pieces to this complex assessment puzzle, if you will. And I think in terms of really moving that transition, regulatory decisions require this cohesive end-to-end evidence frameworks that are familiar to them. And so I think this also touches a little bit on the inertia and how do we communicate an uncertainty that regulators are less familiar with, that we haven't yet harmonized. And I think we're having a harder time to communicate what these different uncertainties are when looking at NAMs as compared to the uncertainties that we're used to managing and communicating in the context of the animal studies. So I think this really comes down to a little bit thinking about the harmonization, but also how do we work towards the standardization and how do we define a method that is considered to be either validated or the qualification type step that Lorna spoke to. So I think it's really all of these beautiful puzzle pieces. How do we put them together, demonstrate that really addresses the needs that the animal studies have been addressing?
Yeah, and I'll just finish off with the related views here as well. Try not to repeat what the other panelists have said already, but down at the bottom there, funding is evident. And, I'm based in the United Kingdom, and we know that funding for, how we start to, gather some of the data on new models that are developed or models, then as Tara has reminded us, some of these are not new, right? So there's a lot of data out there already, but maybe there's still some gaps to fill in those data packages to allow us to claim validation and eventually qualification. The funding picture is very challenging there because many organisations don't really appreciate the value in generating those data points and encourage this sort of blue sky, the moonshot type thinking. So it'd be fantastic if we could perhaps share wisdom to continue the education and the awareness in amongst our funding organisations. When we're looking at building these models or filling in some of these gaps. But likewise, I work for a company that's privately held. It relies on investor investments into the organisation, and again, they need to be educated on. Well, we are making tremendous progress. Please keep funding us. So I think funding is a really pertinent point to be raised here.
Yeah, and speaking of funding, the road ahead, we mentioned that here on this slide as well. So those are all great points that you all brought up. And actually, now that we're here, we are getting some great questions in the Q&A. So I will ask some. I will return back to the question. So just keep it top of mind about what the audience can do to support the transition. But I do want to get to some of these questions. And just a reminder to the audience, if you have questions, to submit, the Q&A box is towards the bottom of your screen. There was one question here that asks, with all of the good news that Troy shared regarding shifts, why do the numbers of animals used in research in Canada keep increasing?
I mean, I can speak to that briefly. If we look at the statistics from the Canadian Council on Animal Care, the different categories of animal use. So there is basic research looking at not medical, it's understanding basic biology. That is the number one use or proportion where animals are being used. It's not cure for cancer, it's not trying to crack diseases specifically. That is, I don't want to diminish basic research, but this is an area where it happens because somebody funds it. And the decision on where public funding, it's largely public funding by Canadian research funding institutions, that a decision could be taken similar to what the US NIH has done with making an announcement, making an internal policy. You don't need laws, you don't need politicians, you just crack a policy that says we are not going to do any animal-specific funding calls any longer. We are going to do ring-fenced funding for specific non-animal method development or infrastructure programs. There is some of that. There is an amazing research is being done in this country through government funding, but if we compare that proportionately to the US, there's a program stateside called Complementary, which is 400 million over 10 years. There's nothing that even remotely approximates that up this side of the border. So there is a real missed opportunity because where public funds go absolutely dictates whether there's going to be downstream investment in non-animal methods or animal methods, full stop. And I think there is something to be said also at the level of institutional ethics committees, but funding is upstream. And if it's not funded in the first place, there is no conversation happening at the university or individual laboratory level. So I think that is really where the resources can be targeted. And that is, back to your earlier question, Bianca, what people can be doing is joining webinars, joining conversations like this to become informed, because the perception that politicians in particular have, they look at public opinion polls and sure, 90+% are opposed to animal testing for cosmetics. It's cruel, it's frivolous. If it's from medical research, it's it's viewed as a necessary evil. And as long as policymakers perceive that the public has conditional interest or support rather than a nuanced and sophisticated understanding of the issue and an ability and a willingness to engage and ask the hard questions, then, I think that is really a pivot point. And we see that increasingly happening with undercover investigations, like what happened over the summer with some institutions in Ontario. There, there is a large public outcry. There was recently, just this week, an investigation released on animal use in the UK. And look at those images, look at the reality, because I think seeing is believing, and having the conversation with friends, helping, using our voice to share facts, not fake news, but really the reason we have this panel today is to have authoritative voices who are sharing the truth. And I think the more people who are acquainted with that and who are able and willing to have these conversations, that's what's shifting the paradigm.
Great. Thank you, Troy. There is another question here, and I think this may have been answered, but I'll ask anyway in case anyone has anything to add. An attendee asked, to what do you attribute ethical concerns becoming mainstream? I'm relieved it's the case, but wonder what has finally begun to turn the tide. And I think you kind of touched on that, Troy, about public opinion and that. But if anyone else has anything to add, I'll open the floor to the panelists.
I mean, I think I'm just going to reiterate the last few things that Troy was describing there. I think it's the awareness that these technologies exist, and it's that, the speaking of the truth and sharing the message. And, I'm just going to divert temporarily, so please stay with me. Just honestly, I'll just do a few seconds. That the image on the left-hand side of the slide we're looking at is the emulate bone marrow chip, and I was privileged to be part of the development of that model. Many people may not know, but those chips have actually just been on the Artemis 2 mission. So they've been right round the world, right round the moon and back down again. And we had our marketing team just monitor the reach that news got. And we through the metrics that they do, and I'm not a marketeer, but because of the outlets that covered that, so we had CNN, we had the BBC, we had Nature, we had Science, all cover this about the importance of these chips going around the moon. It reached 4.9 billion people. That's half the world. Right? Half the world now know an organ chip. Now they might not know what that was, but they know that it went up in with the astronauts and went round the moon. So, that reach that we can get there is unprecedented. Might be the only time in my lifetime that kind of reach is happening, but that's what I live for. I live for, more and more people really understanding what these technologies can deliver. So maybe we need more astronauts going to space with them with organ chips and other models, but hopefully that just is a parallel point.
Yes, that's great. Thank you both for that. I will move into another question here. This is about universities. This is a university related question. In universities, many departments are slow in getting away from animal experiments. What needs to be done in education to facilitate a change in the right direction? And I think, again, we've probably touched on this with some common themes, but I will put this over to the panelists if there's anything else you'd like to add.
If we look at animal use in education specifically, I mean, this is one of those places where vet schools around the world and, in certain economies have eliminated the harmful use of animals full stop. And I mean, and for decades in some places. So if you can teach a veterinarian to do advanced surgery, to do all of the clinical work with animals, what is the excuse if you go backwards to vet techs, if you go backwards to PhD, undergrad, back to the dissection of a frog in grade 7. I mean, it's a bad habit, but I think this is a place where there's really no conversation to be had around educational or pedagogical merit. This is, I don't care if I'm the educator. I'm familiar with this. I have the budget for this. I've always done it this way, and no one's stopping me. So I think it's an accountability problem. And this is one of those places where I think the Canadian Council on Animal Care can and should be much more proactive in ensuring that when you say in your guidelines that animals should only be used after an investigator's best effort to find an alternative failed, this is one place where the alternatives exist. There are large databases of them, and they're being utilized by those who are motivated. Those who are not motivated need some consequences, whether that means, if a university is failing to use them, they could have their certificate of good animal practice suspended by the CCAC. Secondary institutions are not CCAC accredited, but again, those are boards of education or provincial governments could be stepping in with that. But again, I think this is just a Wild West lack of accountability, and that is solvable. But that's one of those areas where, again, what is the consequence to anyone who's not doing the right thing?
To,
I
oh,
think
sorry.
I
Yeah.
just want to add on that as well. And I think there is some responsibility that lies on those of us who are focused in this area and, building the methods and learning the interpretation and, domains of applicability in working in partnership with some of the academic institutions. We have the opportunity, I know, I have the opportunity in some cases to be a guest lecturer in some courses and certainly are introducing the concepts. The students are very interested, and there are a number of academics who, in Canada in particular, are leading in this area and are interested. I think this comes really full circle back to the funding conversation, and that there are opportunities to apply for certain grants in teaching and capacity building areas, but getting the funding to focus particularly in on these types of developments is still challenging. And so I do think that there is interest, and I am seeing that evolution happen. um, I think we need to think about it collectively in terms of how do we provide the tools and support that momentum to introduce some of these technologies and concepts and learnings into the biology courses and toxicology courses where they fit. So, I kind of turn that around that I think we all play an important role in the engagement and the partnership with our academic community to be able to help guide that way forward in terms of capacity building. These are our future, and so I think there is an important investment to be made in translating the information to the students and offering them those opportunities.
And I have a question here that builds on what we're talking about. And it says, I wonder if the structure of university research ethics boards are part of the problem, and that the people approving institutional academic research are by nature colleagues of the person submitting a proposal. Has any thought been given to this? Example, connecting to a level of external approval with funding. Not sure if anyone has anything to add there, but I think that was a good, like a follow-up that came up.
I mean, I think it's the double-edged sword of peer review where it can work really effectively, but there can be you scratch my back, I scratch yours type of interactions. I know many years ago, I was a community representative on assessment panels of the CCAC, so I got to go into a handful of institutions and as part of their 3 to 5 year assessments and speak to animal care committee members. And it was eye-opening for me to have them hear the questions I was asking. And some people were coming up and saying, we don't know what, like the community reps in particular, I don't know how to frame those questions. I'm a fish out of water. I'm a layperson in a room full of scientists. I don't know how to really function in this environment. And even though there are support mechanisms, it's an unfair position to be put in, and people will typically feel, I just can't resist it. So, either I vote for it or I vote against it and I'm outvoted because I'm one of 10 something members. So, and I don't think the answer is to shuffle the deck and have 51% community reps. I think the issue is back to going upstream, because if we're looking at, the current approach is, I'm a researcher who uses mice. I'm going to come up with a mouse experiment. I'm going to submit it to a funding agency, and they fund it or they don't. Versus, I'm looking at the toolbox of NAMs. I have a scientific question. How can I use this modern toolbox of human-based methods to answer the scientific question? That's a fundamentally different pathway to be taking. And for either a peer review at the funding agency level or at an institution to try to take a mouse or a rat or a dog or a monkey experiment and re-engineer and try to, where can you put an organ chip in here? Or where can you, it's too late. So I think we need to go back to even before the research question is formulated. And again, that very much comes down to the way research funding agencies articulate the funding calls to say, this is what's eligible. These are criteria that you need to take into consideration. And it's not about taking an animal experiment, trying to look for replacement, which will fail. And then the best you can do is reduce and refine. That's been the pattern since 1959. And it's very much the status quo today. We need to flip the chessboard over and say, new game.
Great. Thank you so much for answering those questions. Just in the interest of time, I know there are a few other questions that we didn't get to. Our contact information is here. I just wanted to open the floor to the panelists for any final thoughts, any closing words, and if you have the answer to the question, what can the audience do, or anyone watching this recording do to help accelerate the transition? If you have any words of wisdom, please do share. And for any audience members, the contact information you see on the slide, please, you can reach out to the email there, and we will direct any questions that we weren't able to answer to panelists, and we can answer those offline for you. But I'll just turn it over to the panelists for a final word.
I don't mind
I can start.
starting. Oh, with Tara and
Go
I.
ahead, Lorna.
All right, thank you. My one ask would be for the audience to remain curious. We'll do our best, or certainly if I'm allowed to speak on behalf of Troy and Tara here, we're evangelists. We're very happy to always talk and share the best of our knowledge and data and so on and so forth. But I would encourage the audience to be curious, to seek that information themselves, to ask questions and challenge us back as well. It's a conversation, and I think Troy said it very well there. It's time to turn the chessboard over and start a new game. So be curious. That'd be my final words.
I guess my final perspectives would certainly be that this is a global effort. And I think success really is based on partnerships and collaborations. And so all perspectives are important. And as we move this bar together, I think it's really important that we're learning together, developing together, and being able to identify the path to the implementation and alignment internationally. And so I guess encourage everybody to look for those partnerships and to continue to move the conversation forward. As difficult as it is in certain aspects, I think it's the difficult questions that are the interesting questions. And so that links very nicely to the be curious, but let's do it together. And thank you very much for the opportunity to be here. I always learn from all of you. Thank you.
I guess I'll just close by saying, without aging myself, I mean, we used to measure progress one retirement at a time. It used to be so slow and so painful. And looking to where we are today, it is almost whiplash to see how quickly things are moving. So despite the pain points, I guess it's just a relative reflection on we went from having big ideals and driven mainly by ethics. Science has now caught up, and now there's momentum, there's capacity, and it is becoming so mainstream. I mean, the fact that we have panels like this today and all the time that bring industry, government regulator, animal welfare together to share very common perspectives. The amount of sunlight between the different perspectives is so small today that it really is a question of how quickly rather than if we're going to get there. And that really warms my cynical heart because I think we are moving at record pace. Now it's just how do we keep going? And I think having the public continue to engage with this, realize it's not just cosmetics. We didn't end this years ago. It is still ongoing, but it is achievable. And it's not just the experts who need to do the work, it's the everyday person who needs to have a better understanding, be able to communicate this with friends, family, lawmakers. We see in countries where this topic becomes an election issue, things happen, things move quickly, but it's not an election issue in Canada or North America, and that needs to change. But that's down to each and every one of us doing our part. And so thank you all for being here today. And yeah, very much thank you to Animal Alliance for organizing this panel and to Lorna and Tara for joining us.
Yeah, thank you very much.
Yes.
Thank you.
Thank you. We appreciate everyone who joined us today, everyone who's watching, who registered for the recording, and to our wonderful, amazing panelists for being here. For everyone watching, you will receive resources, links, and a copy of this recording. And please reach out to us if you have any questions, and we will get answers for you. So thank you so much, and everyone have a great day.
Thank you.
You
Bye.
too. Thank you.